Hello everyone, I am Mohammed Hamama, and this is your ASCP preparation camp. In this camp, we will go through each topic on the ASCP lecture list. In today's video, we will be discussing anemias caused by defects of DNA metabolism. Megaloblastic anemia, which falls under the category of macrocytic anemias, is caused by impaired DNA synthesis. The reduction in cell divisions leads to the distinctively large cells found in the bone marrow. To comprehend the root cause of megaloblastic anemia, a review of DNA synthesis is necessary, with specific emphasis on the functions of folic acid, folate, and vitamin B12, cobalamin. First we will talk about the physiologic roles of vitamin B12 and folate. The physiological roles of vitamin B12 and folate are essential for human health. Vitamin B12 is a coenzyme in two biochemical reactions in humans. One reaction involves the conversion of methylmalonyl-CoA to succinyl-CoA, which requires vitamin B12 as a cofactor. The other reaction involves the transfer of a methyl group from 5-methyltetrahydrofolate to homocysteine, which generates methionine and requires vitamin B12 as a coenzyme. This reaction links folate and vitamin B12 coenzymes and is necessary for normal erythropoiesis. Vitamin B12 deficiency leads to impaired activity of methylmalonyl-CoA mutase, resulting in a high level of serum methylmalonic acid, which is useful for diagnosing vitamin B12 deficiency. Folate is a general term used for any form of vitamin folic acid, with folic acid being the synthetic form in supplements and fortified food. Folates transfer carbon units in the form of methyl groups from donors to receptors, playing an essential role in the metabolism of amino acids and nucleotides. Folates have an important role in DNA synthesis, and the process involves the transfer of a methyl group from 5-methyltetrahydrofolate to homocysteine, which converts it to methionine and generates tetrahydrofolate. This reaction requires vitamin B12 in the form of methylcobalamin as a cofactor. Tetrahydrofolate is then converted to 5,10-methylene tetrahydrofolate, which is then used to synthesize deoxythymidine monophosphate, a building block of the DNA molecule. Regeneration of tetrahydrofolate requires an additional 5-methyl-THF from the plasma. Folates are rapidly polyglutamated upon entering the cell, which is required for the retention of tetrahydrofolate and promotes the attachment of folate to enzymes. A deficiency of folate leads to impaired cell replication and other metabolic alterations. Defect in megaloblastic anemia due to deficiency in folate and vitamin B12. The production of thymidine nucleotides for DNA synthesis is hindered when either folate or vitamin B12 is deficient. Folate deficiency has a direct effect on thymidine nucleotide production by preventing the methylation of deoxyuridine monophosphate. Vitamin B12 deficiency has an indirect effect by preventing the production of THF from 5-methyltetrahydrofolate, which causes the folate trap to occur. The folate trap is characterized by the accumulation of 5-methyltetrahydrofolate, which cannot supply the folate cycle with tetrahydrofolate. Additionally, homocysteine accumulates when either folate or vitamin B12 is deficient, as methionine synthase requires vitamin B12 as a cofactor. In a state of decreased thymidine availability, uridine is incorporated into DNA, leading to the formation of single-strand breaks and double-strand breaks when excisions at opposing DNA strand sites coincide. Repeated DNA strand breaks cause fragmentation of the DNA strand, resulting in non-functional DNA and incomplete DNA replication. Ineffective hematopoiesis occurs due to the halted cell division, leading to cell lysis or apoptosis of many erythroid progenitor cells within the bone marrow. The dependency of DNA production on folate is utilized in cancer chemotherapy. The remaining erythroid cells are larger than normal and have immature-appearing nuclei compared to the cytoplasm due to cell cycle delay, the prolonged resting phase, and arrest in nuclear maturation. Reduced synthesis of histones is responsible for morphologic changes in the chromatin of megaloblastic erythroid precursors. RNA function is not affected by vitamin B12 or folate deficiency since RNA contains uracil instead of thymidine nucleotides. The slower maturation rate of the nucleus compared with the cytoplasm is called nuclear cytoplasmic asynchrony. The accumulation of cells with nuclei at earlier stages of development and cells with increased size and immaturity leads to the pathognomonic appearance of erythroid cells in the bone marrow in megaloblastic anemia. Pancytopenia is also evident due to ineffective hematopoiesis affecting all three blood cell lineages, with certain distinctive cellular changes seen in laboratory diagnosis. Other causes of megablastosis. There are additional causes of megaloblastic erythrocytes besides vitamin B12 and folate deficiency. Myelodysplastic syndrome can cause dysplastic erythroid cells with megablastoid features, but these can be differentiated from megaloblastic RBCs caused by vitamin deficiencies by delayed cytoplasmic and nuclear maturation, as well as other morphologic differences. Congenital dyserythropoietic anemia types 1 and 3 can also cause nuclear cytoplasmic asynchrony and megaloblastic RBCs, but these can be distinguished from acquired causes of megablastosis by clinical history and morphologic differences. Acute erythroid leukemia and reverse transcriptase inhibitors used to treat HIV infections can also cause megaloblastic changes, but these are due to acquired or inherited mutations in progenitor cells or interference with DNA synthesis and are unresponsive to treatment with vitamin B12 or folic acid. Systemic manifestations of folate and vitamin B12 deficiency. Lack of folate or vitamin B12 can cause megaloblastic anemia and its related symptoms such as fatigue, weakness, and shortness of breath. Deficiencies can also cause alimentary tract symptoms including glossitis, gastritis, nausea, or constipation. Vitamin B12 deficiency may also cause neurologic symptoms such as memory loss, numbness, tingling in toes and fingers, loss of balance, and impaired walking. Folate deficiency may increase the risk of cardiovascular disease and cause depression, peripheral neuropathy, and psychosis. Folate deficiency during pregnancy can result in neural tube defects such as spina bifida in the fetus. Adequate folate levels are particularly important for women of childbearing age because they may be unaware of pregnancy during the early crucial weeks of fetal development. Causes of vitamin deficiencies. Vitamin deficiencies can occur due to a variety of reasons, including insufficient supply, impaired use, or excessive loss. Folate deficiency can be caused by any of these mechanisms. Folate deficiency. Inadequate intake of folate can lead to deficiency. Folate is found in many foods, including leafy green vegetables, dried beans, liver, beef, fortified breakfast cereals, and some fruits. However, overcooking of food can reduce the nutritional value of folate. Increased need for folate occurs during pregnancy, lactation, and growth. Infants and children require more folate for their growing bodies, and pregnant women must supply folate for both themselves and their developing fetus. Impaired absorption of folate can be caused by intestinal disease, such as sprue and celiac disease, as well as a rare genetic deficiency of a folate transporter protein. Food folates must be hydrolyzed in the gut before absorption, and only about 50% of what is ingested is available for absorption. Impaired use of folate can be caused by numerous drugs, such as anti-epileptic drugs, which can result in macrocytosis with megaloblastic anemia. Folate inhibitors like methotrexate are used in some anti-cancer drugs. Excessive loss of folate through the kidney is usually not a cause of deficiency, but patients undergoing renal dialysis can lose folate in the dialysate and may require folic acid supplementation to prevent megaloblastic anemia. Vitamin B12 deficiency. Vitamin B12 deficiency can occur due to inadequate intake, increased need, or impaired absorption. Strict vegetarians who do not consume animal products may be at risk of deficiency as vitamin B12 is only available from animal sources. The increased need can occur during pregnancy, lactation, and growth. Impaired absorption. When we eat food containing vitamin B12, it is released from food proteins in the acid environment of the stomach and is then bound by a salivary protein called haptocorin, also known as a protein or transcobalamin I. In the small intestine, vitamin B12 is released from haptocorin by pancreatic proteases and then bound by an intrinsic factor, which is produced by gastric parietal cells. Vitamin B12 binding to intrinsic factor is necessary for its absorption by ileal enterocytes possessing receptors for the complex. Vitamin B12 then enters the circulation bound to transcobalamin and is released into cells where it is metabolically active. The absorption of vitamin B12 can be impaired due to various reasons like a failure to separate vitamin B12 from food proteins or haptocorin, lack of intrinsic factor, malabsorption, and competition for available vitamin B12. If the body fails to release vitamin B12 from food proteins due to hypochlorhydria or atrophic gastritis, it results in foodcobalamin malabsorption. Long-term use of histamine 2 receptor blockers, proton pump inhibitors, and gastric bypass surgery can also cause foodcobalamin malabsorption. Lack of gastric acidity or trypsin due to chronic pancreatic disease can prevent vitamin B12 absorption by keeping it bound to haptocorin in the intestine. Lack of intrinsic factor is another significant cause of impaired vitamin B12 absorption and is commonly due to autoimmune diseases like pernicious anemia or loss of parietal cells with Helicobacter pylori infection, total or partial gastrectomy, or hereditary intrinsic factor deficiency. Pernicious anemia is an autoimmune disorder that is caused by the destruction of gastric parietal cells by autoimmune lymphocytes, resulting in a reduced secretion of intrinsic factor, which impairs the absorption of vitamin B12. The disease was called pernicious because it was fatal before its cause was discovered. Pernicious anemia mostly affects people over the age of 40, but it can also occur in children. Patients with pernicious anemia have an increased risk of developing gastric tumors. The autoimmune response in pernicious anemia also produces antibodies to intrinsic factor in gastric parietal cells, which can be detected in serum. The most common blocking antibody to intrinsic factor inhibits the formation of the intrinsic factor vitamin B12 complex, preventing the absorption of vitamin B12. The absence of intrinsic factor can be detected using the Schilling test, but safer diagnostic tests are now available. Please don't forget to subscribe to our channel, activate notifications to get our new videos. If you like our content, please press the like button, and share the video with your friends. If you have any questions, leave a comment below. Laboratory diagnosis. Laboratory diagnosis of megaloblastic anemia involves both screening and specific diagnostic tests aimed at identifying the particular vitamin deficiency and its cause. To screen for megaloblastic anemia, 5 tests are commonly used, including the complete blood count, reticulocytes count, white blood cell manual differential, serum bilirubin, and lactate dehydrogenase. A CBC and reticulocytes count are useful for detecting slight macrocytosis, which is often an early sign of megaloblastic anemia. Patients with this condition typically have reduced hemoglobin and hematocrit levels, pancytopenia, and reticulocytopenia. The MCV is usually 100 to 150 femtoliter and commonly greater than 120 femtoliter, but other conditions like iron deficiency or inflammation can prevent macrocytosis. The MCH is elevated due to increased cell volume, but MCHC is typically within the reference range. The RDW is elevated, and peripheral blood film may show oval macrocytes and hypersegmented neutrophils with 6 or more lobes. The severity of the anemia is often severe when first detected, with hemoglobin values of less than 7 or 8 grams per deciliter being typical. Erythroblasts with megaloblastic nuclei, including an occasional promegaloblast, may appear in the peripheral blood when the hematocrit is less than 20%. Other morphologic changes may include the presence of teardrop cells, RBC fragments, and microspherocytes. These smaller cells further increase the RDW. Nucleated RBCs, Howell-Jolly bodies, basophilic stippling, and Cabot rings may also be observed. White blood cell manual differential. Hypersegmentation of neutrophils is a distinctive feature of megaloblastic anemia that is typically present early on in the course of the disease and may persist for up to 2 weeks after treatment begins. When noted in the results of a white blood cell manual differential, hypersegmented neutrophils are considered a significant finding that requires a consistent reporting rule, since even healthy individuals may occasionally have one. One reporting rule is to report hypersegmentation when at least 5 5-lobed neutrophils are present per 100 WBCs, or at least one 6-lobed neutrophil is noted. Some labs calculate the mean lobe count on 100 neutrophils, and in megaloblastic anemia, the mean lobe count should be greater than 3.4. The cause of hypersegmentation is not yet fully understood, despite ongoing research. However, looking for neutrophil hypersegmentation on a peripheral blood film is a cost-effective and sensitive screening test for megaloblastic anemia. Bilirubin and lactate dehydrogenase levels. The levels of bilirubin and lactate dehydrogenase are elevated in patients with megaloblastic anemia, which is considered a type of hemolytic anemia. Ineffective hematopoiesis, where many red blood cell precursors die during division in the bone marrow, results in a decrease in reticulocytes in the peripheral blood. The increase in the levels of total and indirect bilirubin and lactate dehydrogenase in the serum is due to hemolysis of RBCs. To confirm a diagnosis of megaloblastic anemia and identify its cause, additional testing is required, given the presence of macrocytic anemia, moderate to marked pancytopenia, reticulocytopenia, ovomacrocytes, and hypersegmented neutrophils, along with elevated levels of bilirubin and lactate dehydrogenase. However, the diagnosis may be challenging if there are coexisting conditions, such as iron deficiency, which can obscure the classic findings. In most cases, hematologic abnormalities do not appear until the vitamin deficiency is well advanced. Specific diagnostic tests. Bone marrow examination. Bone marrow examination is now less commonly used for diagnosing megaloblastic anemia due to the availability of modern tests for vitamin deficiencies and autoimmune antibodies. However, it remains the gold standard test for confirming the megaloblastic appearance of developing red blood cells. Unlike macrocytic anemia, megaloblastic anemia is characterized by distinct morphological changes in developing red blood cells. The cytoplasm matures as expected, becoming increasingly pink as hemoglobin accumulates. However, the nucleus lags behind and appears younger than expected for the degree of cytoplasmic maturity, which is most evident at the polychromatic normoblast stage. The bone marrow is hypercellular, with a myeloid to erythroid ratio of approximately 1:1, reflecting increased erythropoietic activity. However, the hematopoiesis is ineffective, resulting in peripheral pancytopenia due to apoptosis of cells in the marrow. White blood cells are also affected in megaloblastic anemia and appear larger than normal, especially in metamyelocytes and bands. This creates giant metamyelocytes and bands. The changes in megakaryocytes are inconsistent, with no consistent increase or decrease in their numbers. The diminished lobulation, when present, is difficult to assess. Assays for folate, vitamin B12, methylmalonic acid, and homocysteine. While bone marrow examination is the confirmatory test for megablastosis, its invasiveness and expense make other tests, such as assays for serum levels of folate, vitamin B12, methylmalonic acid, and homocysteine, more commonly used. However, these tests can produce false results due to interferences, so reflexive testing for methylmalonic acid and homocysteine can increase diagnostic accuracy. RBC folate levels may also be measured but have less than optimal sensitivity and specificity. In laboratories, some conduct a reflexive assay for methylmalonic acid if vitamin B12 levels are low. Homocysteine levels are affected by deficiencies in either folate or vitamin B12, and elevated levels can be measured in plasma or serum by different methods. Gastric analysis and serum gastrin. To confirm achlorhydria, which is often seen in pernicious anemia, gastric analysis can be performed. However, this condition can also occur due to natural aging or other factors, and therefore, the absence of gastric acid is not diagnostic of pernicious anemia unless other causes of vitamin B12 deficiency have been ruled out. The hydrogen ion concentration is measured by pH measurement. In cases of gastric achlorhydria, serum gastrin levels can be significantly increased. The measurement of serum gastrin is conducted using immunoassay techniques, including chemiluminescent immunometric assay. Antibody assays. Most patients with pernicious anemia have detectable serum antibodies to intrinsic factor and parietal cells. Immunoassays can be used to detect intrinsic factor blocking antibodies, while indirect fluorescent antibody techniques or enzyme-linked immunosorbent assays can detect parietal cell antibodies. Although anti-IF antibodies are highly specific and confirmatory for pernicious anemia, their absence does not exclude the condition. On the other hand, the test for parietal cell antibodies is not clinically useful for the diagnosis of pernicious anemia due to its nonspecificity. Other tests. Holotranscobalamin is the active form of vitamin B12 can be measured by immunoassays that use monoclonal antibodies. Although recent studies show that holotranscobalamin is not very specific in detecting vitamin B12 deficiency, and it is not recommended for routine clinical testing. The deoxyuridine suppression test works on the principle that normal bone marrow pre-incubated with deoxyuridine can suppress the subsequent incorporation of labeled thymidine into DNA because normal cells can methylate uridine into thymidine. However, in patients with vitamin B12 or folate deficiency, suppression is abnormally low. By adding vitamin B12 or folate to the test cells, one can determine whether the inadequate suppression is caused by vitamin B12 or folate deficiency. However, this test is complex and impractical for clinical testing due to the necessity of using bone marrow tissue. When a patient is found to have vitamin B12 deficiency, a stool analysis for eggs or proglottids of the fish tapeworm D. latum may be performed as part of the diagnostic workup. Macrocytic non-megaloblastic anemias. Macrocytic non-megaloblastic anemias are characterized by the presence of macrocytic anemia in which DNA synthesis is not impaired. Usually, the macrocytosis is mild and the mean corpuscular volume, MCV, ranges from 100 to 110 femtometers and seldom goes beyond 120 femtometers. Patients suffering from macrocytic non-megaloblastic anemias do not have hypersegmented neutrophils or oval macrocytes in their peripheral blood or megaloblasts in their bone marrow. Macrocytosis may be physiologically normal in newborns or caused by pathological conditions such as liver disease, chronic alcoholism, or bone marrow failure. Reticulocytosis is a frequent cause of macrocytosis. Thank you for completing the video. Remember to ask for ASCP short notes, and don't forget to subscribe. Bye.